Hello everybody. Today I am going to discuss about a very interesting topic, which is the nervous system. So in this session we will be discussing about the following aspects of the nervous system. So first we will be seeing the subdivisions of the nervous system, followed by the components of central and peripheral nervous system. Then we will be discussing about the structure of a typical spinal nerve, the structure of a neuron, classification of neurons and neuroglia with examples, what is a synapse, how a synapse works, and various types of synapse. Then autonomic nervous system, and finally we are going to see some few aspects of the neuronal degeneration and regeneration. So basically, what is special about the nervous system? So it is a controlling and coordinating system of our body. So it controls mainly the activities of our body. How do we respond to the stimulus which we actually receive externally from the environment, and also from the stimulus which we receive within the body, internal from our internal environment, and then it coordinates all these information which is receives. So for this we need a special type of cells which are actually called as the neurons. The neurons are excitatory cells which can sense, then after sensing it processes and then it responds, that means taking appropriate action based on the information which it has received. So that means it is constantly receiving information, that is impulses, with respond to the internal and external environment, that is from within or outside the body. Then of course it processes the impulses, so that means it checks whether everything is right or wrong, or something has to be done regarding it, and then it gives proper commands, thereby sending adequate responses to the effector organ. So it might be a viscera or a muscle, and so other structures in our body. Not only that, it also stores adequate information from the past experiences, and it synchronizes with endocrine system for maintaining the homeostasis, that is the internal environment of the body. Okay. So basically the nervous system can be classified into two parts. One is actually called as the central nervous system, which comprises of the brain and the spinal cord. The peripheral nervous system is the nerves which are actually arising from the brain and the spinal cord. So you have certain nerves which are emerging from the cranial nerves, they are called as, and spinal nerves are nerves which are emerging from the spinal cord, and their associated ganglia. So I will come to what is ganglia after some time. Then apart from the central nervous system and peripheral nervous system, you also have another small subdivision which is actually called as the autonomic nervous system, with its two components, that is the sympathetic and the parasympathetic component. So basically the nervous system is classified into central nervous system, peripheral nervous system, and then autonomic nervous system. Now coming to the central nervous system, so the basic components of the central nervous system. So it is the highest center for control of sensory and motor functions. So whatever the information which it receives from the periphery or from outside the body, from the environment, everything has to be actually transferred to the central nervous system for processing. And from there what happens is appropriate commands are given or information is actually again travels down, which also has to originate from the central nervous system. So it is not only performing the sensory functions or motor functions, it is also a seat of higher functions like your intelligence, your memory, your personality, your thinking, your way of perception, emotions, intelligence and learning. Everything is actually controlled by this central nervous system. So basically the central nervous system comprises of, this is actually called as the hindbrain, that is your cerebellum, medulla and pons. Put together, which is actually called as the rhombencephalon. And next is actually called as the midbrain. It's called as midbrain because it is present between the two cerebral hemispheres. So that is why it is actually called as the midbrain. Consists of this midbrain alone, and it is called as the mesencephalon. The next one is actually called as the prosencephalon, which comprises of forebrain, this large one called as the cerebrum, prosencephalon, and one more component is actually called as the diencephalon. So the diencephalon and the prosencephalon put together form the forebrain. The diencephalon is actually a sensory relay center. It's only a relay center, but again from here it has to go to the cerebral cortex, all highest control for sensation and highest control for motor activity, and as I told you, for all other higher functions such as intelligence, memory, learning and emotions. Okay, so these are the basic components of the central nervous system. So forebrain, midbrain and hindbrain. Next, apart from the brain, you also know the central nervous system also comprises of this long cord-like structure which is called as the spinal cord. So what is spinal cord? So spinal cord can be simply explained as thick bundle of nerve fibers and very well situated within the vertebral column, and it is the main pathway. So whatever the sensations which we receive passes through this, and what are the motor commands also has to come through it. So it can simply be called as the information pathway. Mainly made up of axons. Neurons are also there, but mostly populated with axons, and these axons are actually called as tracts. Okay. So there are tracts which are actually coming down. They are called as the tracts or motor tracts, descending tracts. There are tracts which convey sensation, sensory information, so they are called as ascending tracts. So spinal cord mainly consists of tracts, and not only that, it also has various centers, reflex centers. So reflex centers mainly for actions such as micturition, defecation, respiration, and also cardiac centers are also present in this spinal cord. So it is a connection between the nerves, that is the central nervous system, and the peripheral nervous system. So it connects the central and peripheral nervous system, this spinal cord. Now coming to the peripheral nervous system. Peripheral nervous system means it is mainly made up of nerves. So nerves which are directly arising from the brain is actually the cranial nerves. There are 12 pairs, 12 on each side, so naturally 24 nerves. So here you can see the names of these nerves. It is not necessary to memorize the names of all these nerves at this stage, because these are only introductory sessions which are actually dealing. So it is called as olfactory, optic, oculomotor, trigeminal, trochlear, abducens, facial, vestibulocochlear, glossopharyngeal, vagus, accessory and hypoglossal nerves. So they are called as cranial nerves because they directly emerge from the brain. Then you have the spinal nerves. There are actually 31 pairs of spinal nerves, which can again be subclassified into 8 from the cervical region, 8 nerves, 12 from your thoracic region, 5 from your lumbar region, 5 sacral nerves and 1 coccygeal nerve. So actually there are 31 pairs of spinal nerves which are emerging from the spinal cord. And of course I already told you there is some ganglia associated with these nerves. So cranial nerves might have certain ganglia. In the same way, the spinal nerves are also certain ganglia associated with it. Okay. So this peripheral nervous system, through which it provides sensory input, not only provides sensory input, again it also carries back or downwards the motor commands to the target areas. So now we are going to see the structure of a typical spinal nerve. So this is actually called as a typical spinal nerve, arising from any segment, like for example, 8 cervical spinal nerves are there. So let us assume we are discussing about the structure of C3 or third cervical spinal nerves. Now each spinal nerve is connected to the spinal cord by means of rootlets. So they are actually called as the ventral and dorsal or anterior and posterior rootlets. Okay, this is actually called as the anterior and posterior rootlets. Okay, so now what happens is it has got a dorsal root and it has got a ventral root. So here I am able to see a nerve has got a ventral root and this is actually called as the dorsal root. When you look at the dorsal root, again it shows a swelling which is actually called as the dorsal root ganglion. So dorsal root ganglion is actually seen in the dorsal root. Okay. Now when you look at it, now both the roots, the dorsal root and the ventral root, they unite and form a typical spinal nerve. So this is a typical spinal nerve which you are able to see. They are formed after the union of the dorsal root and the ventral root. Dorsal root carries a ganglion. There is a ganglion which is present, which I have already told you. This is called as the dorsal root ganglion. So this union of both the roots takes place at the intervertebral foramen. So that is the space present between the two vertebrae at the sides, and now they are called as a typical spinal nerve. The dorsal root is sensory in nature, whereas the ventral root is motor in nature. So it carries afferent sensations, afferent impulses, and these transmits efferent impulses, that is motor, to the muscles or any target organs. Now after these two roots unite, now it is called as a typical spinal nerve, which is a combination of both sensory and motor fibers. So a typical spinal nerve is always a mixed nerve. Now what happens is this spinal nerve divides into two. Here you are able to see it divides into two. This is called as the dorsal primary rami and this is called as the ventral primary rami. Okay. So these are dorsal and ventral roots, not to be confused with this primary rami. This is actually called as dorsal primary rami and this is called as the ventral primary rami. Okay. Now what happens is the ventral primary rami of the cervical region, lumbar, sacral, they actually unite with the other nerves and they form what is called as the plexus. So again, plexus we are going to see in detail in the upper limb, whereas the ventral primary rami of this region, the thoracic region, they don't join with the cervical nerves above or the lumbar or sacral nerves, and they pass independently, and they do not form plexus or involve in the formation of plexus. So naturally, these nerves only can be considered as a typical spinal nerves, and even in that, the first thoracic nerve also we can exclude. Only starting from T2 to T11 are typical spinal nerves. So they pass independently, T2 to T11. Now, what are dermatomes? So dermatomes is the area of skin. So you are able to see the mapping of dermatomes, the ventral side and the back posterior side of our body. So you can see the area of skin which is actually supplied by a single spinal nerve, that is actually called as the dermatomes. The cervical, how they supply the skin. Then the thoracic, so how they are innervating the skin. Lumbar and sacral. So color schema is given here for you to understand. And even certain important areas are given, like your nipples is supplied by the T4 spinal nerve, your xiphoid process by the 7th thoracic spinal nerve, umbilicus by the 10th thoracic spinal nerve. Okay. Now, so that means an area of skin supplied by a single spinal nerve. Okay. Now, suppose if that skin is damaged or cut in that area, do we lose sensations completely? It is not true. We don't lose sensation. That means the dermatomes, they overlap very frequently. So because of the overlapping of the dermatomes, and more commonly it is seen in the limbs, the overlapping of the dermatomes. So because of overlapping only, what happens is we don't lose sensation so easily. Okay. Now we are going to discuss about the functional unit of the nervous system, which is actually called as the reflex arc. Now, what is a reflex arc? It is the basic functional unit which performs an integrated neural activity. The components of reflex arcs is a receptor. So in this case, let us understand the skin. Then a sensory neuron, which is going to carry the sensations from the skin. And then what happens is, this sensory neuron is going to have connection with the motor neuron, which is going to give command to this muscle. So naturally what happens is, this muscle is going to act and it is going to move that hand away from the sensory impulse if it is an unwanted sensation. Okay. So naturally it consists of a receptor, usually a skin, a sensory neuron, then a motor or efferent neuron, and then affector, example, person. So here you can see a simple reflex arc is actually a monosynaptic reflex. Sensory impulses traveling via the sensory neuron, and then what happens is, from here it will travel to the motor neuron, to the muscle. So this is actually a reflex arc. But in this diagram, what you see is not a simple reflex arc. It is a polysynaptic reflex because of the presence of the interneuron. Okay. So they are called as interneurons. When an interneuron is present between an afferent neuron or sensory neuron and a motor neuron, naturally what happens is, it is actually called as the polysynaptic reflex arc. Okay. So when a sensation is received, naturally it is contacted information, and then the motor neuron makes the muscle contract. So naturally what happens, you can withdraw your hand from the unwanted impulse if it is a pain, or if it is going to be very hot, like temperature sensations, and so on. So simple reflex arc consists of only a receptor, an afferent or sensory neuron, a motor neuron, and an effector organ. Polysynaptic reflex is due to the presence of neurons, that is interneurons, between the afferent and efferent neurons. So a reflex is actually an involuntary motor response following a sensory stimulus. Okay, if somebody suddenly pricks you or somebody suddenly beats you, so naturally what happens is, we try to protect ourselves, and that is actually an involuntary motor. Certain reflexes are also called as conditioned reflex because we can actually condition those reflex. You all would have experienced what is a conditioned reflex. Very simple is driving a two-wheeler or driving a four-wheeler is actually a conditioned reflex. Now, so we have seen about the basic functional components of the nervous system, classification of nervous system into central and peripheral nervous system, components of each central nervous system and peripheral nervous system. Then we have described about a typical spinal nerve, and we have actually discussed about the reflex. Now, we will go to the structure of a neurons. So, neurons, as I already told you, they are special excitable cells, and what happens is, this is the structure of the neuron. It has a body or soma, and it has one long process which is actually called as the axon, and many short processes which is actually called as the dendrites. Okay. The nerve bodies or the soma is mainly forms the grey matter of your brain as well as the spinal cord. Whereas the white matter is mainly formed by the nerve process, and that too especially the axons. Okay. So there are 10 to the power of 6 number of neurons in our cerebral cortex. Okay. Now, what happens is, these neurons don't multiply after certain age, and what happens is, they are constant number. Okay, but what happens is, when we learn new things, when we activate our brain too much, is the synapse is going to increase. The number of synapse is going to increase. So I will discuss about what is synapse after a few slides. So structure of a neuron consists of a nerve body or soma, then it has an axon, then it has got many short process which is actually called as the dendrites. Okay. Now, if you look at the body or soma of the neurons, when these cell bodies are present outside the central nervous system, then you what is actually called as the ganglia. So we have been mentioning about the ganglia few slides back, telling that associated ganglia. So ganglia are collection of nerve cells, that is nerve body, outside the central nervous system. Inside the central nervous system, we call this, if it is a collection, if it is aggregated at one place, then we call it as nuclei. We don't use the term ganglia. So like any other cell, it has got a cytoplasm and all the cell organelles like endoplasmic reticulum, mitochondria, ribosomes. And apart from that, we have some substances which is granular in nature, and that is called as the Nissl bodies. When you use some specific dyes to stain this, then you can see they are all brightly stained bodies and involved in protein synthesis. If the neuron is damaged, then naturally what happens is, these nerve bodies, they disappear or get dissolved, and that process is actually called as the chromatolysis. Arising from the cell body is numerous short processes which is actually called as dendrites, and they branch profusely. If you look into the substance of these dendrites, Nissl bodies are also present inside these dendrites, and neurofibrils are present. If you look at the terminal branches, they show small extensions which are actually called as the dendritic spines. So dendritic spines, mainly what happens is, they increase the surface contact with the other neurons or axons, so that is why the dendritic spines is present. So like how you have the villi which is lining your small intestine. Now coming to the structure of the axon, so it is one long process, a single process only arises from the nerve body, and it divides into small branches but not very numerous, profuse branching like the dendrites which you see here. The starting point of the axon is actually called as the axon hillock, and it is not covered by this myelin sheath. So initial portion of the axon is not myelinated, the point of origin, and then it divides into many small branches, and at the end of the branches has some bulb-like swellings. They are actually called as Bouton dominants. Okay, so the site of synapse actually takes place here. Not only that, neurons also gives collateral branches which runs right angles to the main trunk of the axon. Okay, so we have seen about the structure of the nerve cell, the dendrites, and then the axon. Okay. Next is the cytoplasm is actually called as the axoplasm. Inside the axon, it is called as the axoplasm, mainly consists of neurofibrils, microtubules, and acetylcholine, which is a neurotransmitter. It is a semi-permeable membrane covering the axoplasm, and it's called as axolemma. Like the plasma membrane, it is called as axolemma. Now the neurons might be both myelinated and unmyelinated. Myelinated means when they are covered by a myelin sheath. When they are not covered by a myelin sheath, it is actually called as unmyelinated axons. So myelinated axons, what happens is, they are actually the peripheral nervous system. The nerves of the peripheral nervous system are all myelinated. The non-myelinated axons are mainly seen in the grey matter, in the spinal cord, as well as in your cerebrum. Short unmyelinated axons are mainly seen in the postganglionic fibers of the autonomic nervous system. Very fine sensory fibers, which are actually called as the C fibers, they are 1 micrometer in diameter, less than that. They are all unmyelinated axons. And remember, the tracts of the spinal cord is also not myelinated. Okay. Now let us see how this myelination takes place. The structure which is responsible for the myelination, it is actually called as the Schwann cell. The Schwann cells is responsible for myelination in the peripheral nervous system, so especially in your spinal nerves and cranial nerves. It winds around the axon into concentric layers. You have alternating layers of proteins and lipids. So lipids mainly of cholesterol and phospholipids it is made up of. Certain areas at regular intervals, what happens is this myelination is not absent. They are actually called as the nodes of Ranvier. An unmyelinated segment, it's actually called as the nodes of Ranvier. Then irregular clefts you come across in the myelination, they are actually called as Smiths of Lanterman. They are actually called as Smiths of Lanterman. If you look at the Schwann cell again, it will be covered externally. The Schwann cell will be covered externally by a sheath which is actually called as the neurilemmal sheath. Neurilemmal sheath, and again which will be surrounded by the endoneurium. So neurilemmal sheath interdigitate at the level of nodes of Ranvier. Only deep to the neurilemmal sheath you find the cytoplasm and the nucleus of the Schwann cell. This neurilemmal sheath is very important for the regeneration of the axons. So if the axon is cut, if the neurilemmal sheath is present, then naturally what happens is the regeneration is possible. Okay. Now this neurilemmal sheath is present in myelinated axons and also in unmyelinated axons, but it is absent in non-myelinated axons where regeneration does not take place. So that is a good example of non-myelinated axons is the tracks which forms the white matter of the spinal cord. So that is why what happens is the tracks do not regenerate if they are damaged. So single nerve fiber is actually covered by an endoneurium. The group of nerve fibers which is actually called as the nerve fasciculus is actually covered by perineurium, and all the fasciculus in turn are actually covered by the epineurium. So the epineurium surrounds all the fasciculus, and you are able to see this is a whole trunk of a nerve. So the nerve fiber, the whole nerve, a spinal nerve, if you take, it is covered by the epineurium, and inside you have group of nerve fasciculus which is covered by perineurium, and single one which is covered by endoneurium. Deep to that only you see this axons. Okay, is that clear to you? So next we will see how myelination takes place in the central nervous system. The central nervous system myelination is actually carried out by the oligodendrocyte. So this cell, what happens is it gives out numerous processes which winds around many nerves. So a single oligodendrocyte is responsible for myelination of many nerve fibers, so which makes a bit complicated. So that is why what happens in CNS again, regeneration is not possible because of this. So what is the main function of this myelin sheath? It is mainly responsible for insulation. So it acts like an insulatory material. So thereby what happens is the impulse, unwanted impulse is not transmitted. Then for saltatory conduction, so nodes of Ranvier, what happens is impulse travels from here, then it will jump to the next myelin area, then it will jump. So it is like jump conduction for faster conduction. So that is why it is actually called as the saltatory conduction. Mainly for insulation and saltatory conduction, the myelination is necessary. So in peripheral nervous system it is by the Schwann cell, and in the central nervous system it is by the oligodendrocytes. Okay. So that is how the nerve conduction takes place. This is actually called as the saltatory conduction, which you are seeing here. So now we will go to the classification of neurons. So classification of neurons, you have unipolar neurons, bipolar neurons, pseudo-unipolar neurons and multipolar neurons. Pseudo-unipolar neurons, you have only a single process and at the end it divides. An example is the mesencephalic nucleus of the trigeminal nerve, which is present in the midbrain. Pseudo-unipolar neurons, they are mainly seen in the sensory ganglion and also the spinal ganglia, dorsal root ganglion. We call it as pseudo-unipolar neurons because single cell again divides into a central process and a peripheral process. That is why it is actually called as pseudo-unipolar neurons. Bipolar neurons, where all you see special sensations, your rods and cones, the olfactory epithelium, you see this. Bipolar neurons, cells of the retina and olfactory cells, and also in the cells of the auditory nerves. As I told you, the special sensations are all concerned with these bipolar neurons. Multipolar neurons is a typical neuron which you have seen with one axon on many dendrites. So these are motor neurons which are present in the cerebral cortex. Okay. So that is the classification of neurons: unipolar, pseudo-unipolar, bipolar and multipolar neurons. Okay. Based on the function also you can classify the neurons basically into sensory neurons. Sensory neurons, as I told you, bipolar, special sensations. Pseudo-unipolar neurons which are present in the ganglion, mainly concerned with sensory ganglia, and also in the dorsal root ganglia of the spinal cord. So sensory neurons and motor neurons are multipolar in nature. Okay. So motor neurons, motor ganglia, some of the autonomic ganglia are motor ganglia, they also have multipolar neurons. Between these two is another type which is actually called as interneurons or interventional neurons, which connects the sensory and motor neurons. We have already seen this in the reflex, in the polysynaptic reflex. This interneurons are involved. Neurons can also be classified based on the length of the axon. Golgi type 1 neurons, the axons with more than 2 feet in length, and Golgi type 2 neurons, which we see here, and they are actually shorter axons. Okay, short axons are Golgi type 2 neurons, and Golgi type 1 neurons has got very long axons. Thereby, axons are also can be classified based on their length and size. Type A axons are largest, 1 to 20 micron in diameter. Diameter, not the length, it is the diameter. So the rate of conduction is actually 6 meters per diameter. So that means 120 meters per second. So that is the speed of conduction of type A axons. Type B axons are thinly myelinated autonomic fibers, and the rate of conduction is 3 meters per second. Then you have unmyelinated axons where the rate of conduction is very low, that is 2 meters per second. Okay, the largest axon can also measure around 50 feet in length. Now, how actually impulse travels through this nerve? So briefly, I will just tell you about this nerve conduction. There are two theories which have been postulated with respect to the nerve conduction. One is the membrane theory. So here you are able to see the membrane theory. What happens in the membrane theory is the membrane is actually getting charged as the wave of polarization passes. So slowly what happens is, as the polarization process, the negatively charged and positively charged, they are changing. So this is actually a wave of polarization, and the stimulus travels through the nerve fiber. Okay. Then what happens is, after it is passed, then the membrane comes to its resting position, it gets depolarized. Then what happens is, when you pass a next wave of polarization, then again what happens is repolarization takes place. But there is a gap between one wave of polarization and another wave, and that is actually called as the refractory period. During that period, what happens is the nerve fiber won't get excited. This is called as membrane theory. The next theory, what happens is, okay, there is actually change in the membrane charge, but how actually this change takes place? And that is actually the ionic theory which explains it. There are some gated channels which is present in the membrane, which allows influx of certain ions which pass. So when the polarization is passing, it allows influx of sodium ions. So sodium gated channels are present. So when the sodium ion gets influxed, when the sodium ion increases, then naturally it has to get depolarized, that is to the influx of potassium ions. So influx of sodium ion and efflux of potassium ions, which is responsible for the repolarization and depolarization, and that is how the ionic theory explains. So the rate of conduction takes place based on the thickness of nerve fibers. I told you diameters 1 to 20 diameters. So if the thickness of nerve is more, then what happens is the rate of conduction is more, and the degree of myelination. So myelination, they may be thinly myelinated or thickly myelinated. So that myelin sheath might wind around the axon many layers. So that also increases, and transmission of impulse is usually in only one direction. Whether it might move from upwards to downwards or from downwards to upwards. Okay, so we have seen about The neurons of the nervous system. Now there are also some supporting cells which are actually called as the neuroglia. They are non-excitable. They divide, unlike neurons which usually don't divide. Mitosis, they undergo mitosis. So the neuroglia is a term usually given to the supporting cells in the CNS, that is the central nervous system, and they are mainly astrocytes, oligodendrocytes, ependymal cells and microglia. Schwann cells are also supporting cells which is involved in the myelination of the peripheral nervous system. Then there are also some cells which are actually called as the satellite cells or supporting cells of the ganglia. Around the ganglia you can see these satellite cells. Then there are certain cells or ependymal cells which lines the cavity of your brain. Okay. So in the central nervous system we have already mentioned about 4 types you are able to see: astrocytes, microglia, oligodendroglia and ependymal cells. As the term suggests, microglia, they are very small cells, phagocytic in nature and mainly present along the blood vessels. They are nothing but the monocytes which have transformed themselves into microglia in the central nervous system, and they are actually a mesodermal derivative. So the 3 germ layers of which they are actually derived from the mesoderm. Okay. Whereas the other ones like your astrocytes and oligodendroglia, they are all actually derived from the neuroectoderm only, neuroectoderm. So they are actually derived from the neural tube formation itself. What happens is certain cells, they actually migrate out to various regions and they are actually called as the neural crest cells. So neural crest cells gives rise to so many number of cells which we will discuss later, of which in the central nervous system the supporting cells, mainly the astrocytes and oligodendrocytes, are actually the derivatives of neural crest cells. So microglia, I told you, it's a mesodermal derivative, short with fine processes, and they are phagocytic in nature. So they go, and what happens is anywhere when they injure, neurons are lost, they populate that area and they form a scar tissue. And this is one of the reasons why the regeneration of neurons is not possible in the central nervous system. The next type of neuroglia is the astrocytes. Mainly they are star shaped, so fibers extending in all direction. 2 types of astrocytes can be seen. One is the fibrous astrocytes, which is mainly seen in the white matter along the axons, and other one is actually called as the protoplasmic astrocytes, which is seen along with the nerve bodies, neurons. Okay. So functions of astrocytes is they have a process or end process which they form a perivascular field intimately related to the blood vessels, that is the capillaries, and they contribute to something called as the blood-brain barrier. So what is the blood-brain barrier is? When you allow certain substances you inject into the bloodstream, they are not found in the brain. So that means they allow some selective passage of contents into your brain substance, and that forms the blood-brain barrier. Okay. The components of blood-brain barrier are mainly the endothelium, the internal lining of the blood vessels, the basement membrane where these cells of the endothelial cells are actually resting, and the end feet, the perivascular feet, you call it as the end feet of the astrocytes. 1, 2 and 3, they actually form the blood-brain barrier. Okay. Next type of neuroglia is oligodendrocytes. We have already seen that in the myelination they are actually involved in the formation and maintenance of myelin sheath in the central nervous system. Why it is oligo means less processes, and mainly found in the grey matter and white matter of the central nervous system. Then the next type of neuroglia you see here is the ependymal cells, which are lining the cavities of the brain. So brain and spinal cord as a cavity, they are also called as ventricles. So We will discuss about the ventricles later. They are mainly responsible in exchange of substances, CSF, production of CSF, and they also form the blood CSF barrier. So again there is a barrier between the blood and CSF, because CSF ultimately what happens is it has to go and drain into the venous system. So it also allows some selective passage of substances. Okay. Then the processes of these epidermal cells are also called as Janissines. So we have seen about microglia, astrocytes, which are of two types, protoplasmic astrocytes and fibrous astrocytes, oligodendrocytes, and then epidermal cells. Now coming to what is a synapse. It is a specialized junction between the processes or between two neurons, based on which part of the neuron makes in contact with the other neuron. It is actually classified into axodendritic, between the axon and dendrites of the other neuron, axosomatic, between the axon and the nerve body or soma, that is what is called as axosomatic, or between the two axons it is actually called as axoaxonic. The most common one is the axodendritic type of synapse, which you see here, the first one. And they are mainly excitatory in nature. That means the substance which is traveled inside the neurotransmitter, it is an excitator which excites the other neurons. Okay. The most common type of neurotransmitter is the acetylcholine, which is excitatory in nature. Other type of neurotransmitters are catecholamines, so which is dopamine. Then you also have glutamate, serotonin, and gamma aminobutyric acid. So not all neurotransmitters are excitatory, some are also inhibitory. For example, dopamine is actually an inhibitory neurotransmitters. Okay. So I told you this axodendritic is mostly excitatory in nature, while other types are mostly involved in presynaptic inhibition. So the unwanted excitation has to be controlled. They have to silence the other neurons by getting excited. Okay, so that is why inhibition is also very much necessary. So now we will go into the structure of a synapse. It has a presynaptic membrane, then a space which is actually called as the synaptic cleft, and then what you have is the postsynaptic membrane. So in the presynaptic membrane, the neurotransmitters will be released as vesicles. They will be released into this synaptic cleft. Postsynaptic membrane will have receptors specific for those neurotransmitters, and naturally they will uptake. So that is the structure of a synapse, and most commonly when you look at it, in a type 1 synapse, the postsynaptic membrane is thicker than the presynaptic membrane, whereas in type 2 synapse both the pre and postsynaptic membranes are thin. Type 1 synapse is excitatory, and as I told you, neurotransmitters are acetylcholine. In type 2 synapse, they are mainly, what happens is they are inhibitory in nature, and example is the gamma aminobutyric acid. So here you are able to see postsynaptic membrane gets depolarized once when the neurotransmitter is released. Okay. The action of synapse. So this is released into the presynaptic cleft and it is taken up by the postsynaptic membrane. So structure of synapse mainly consists of presynaptic membrane, synaptic cleft, and postsynaptic membrane. Okay. So far we have seen about structure of neurons, types of neuroglial cells. We have seen about the types of synapses, and now we are coming to some certain components which I will list here. If you look at the peripheral nerves, you know there are motor nerves, there are sensory nerves. The functional components, we call it as, first one is general somatic efferent. So what is a general somatic efferent means? It is mainly motor in nature. General somatic is mainly going to supply to the muscles of your body wall and limbs. General visceral efferent is secretomotor. They are going to involve motor impulses to the glands. Most commonly you can see that your salivary glands, sweat glands and all those things. Special visceral efferent is to muscles which are actually developed from branchial apparatus. I will come to this later. In embryology, we will be knowing about what is branchial apparatus or pharyngeal apparatus. Certain muscles are developed from that, especially the head and neck region. You will see that. General visceral afferent is sensations from the viscera. So pain and sensations and pressure sensations from your organs you receive, heart, lungs, abdominal viscera. General somatic afferent is sensations from your skin, so temperature, pain, touch sensations. Proprioception from muscle and joints, it is actually called as general somatic afferent. Special somatic afferent, you know, this is for special sensations, hearing and olfactory sensations or auditory sensations. But remember, taste sensation is absent because tongue is actually again developed from this branchial apparatus. It comes under special visceral afferent. So if you take any nerve, not all these components will be present in the nerve, whether it is a peripheral nerve, that is the cranial nerves, or spinal nerves. Spinal nerves definitely will not have this special visceral efferent or this special visceral efferent or special visceral afferent. Okay. So one or two components might be present in these nerves. Not all the components will be present in any given single nerve. So coming to the final part of this lecture, a little bit of autonomic nervous system we are going to see. So we have seen about the central and peripheral nervous system. The other subdivision of nervous system is the autonomic nervous system. It consists of two components, sympathetic and parasympathetic. Mainly it is involuntary in nature. It is concerned with your heartbeat, blood pressure, your respiration, rate of respiration, perspiration, sweating and digestion. So mainly it can be influenced even by your thought and emotion. So when you are sad or when you are happy or when you are having some fear, so all those things can influence or activate your sympathetic and parasympathetic nervous system. Now coming to this sympathetic nervous system, it is diffuse and widespread. The effects can be seen throughout the body, and it is actually called as the thoracolumbar outflow. That means the nerves which emerge from the thoracic and lumbar segments of the spinal cord. What happens is they relay into this ganglia, which is actually called as the sympathetic ganglia, which is seen on either side of the spinal cord. Each ganglia is actually connected to a spinal nerves. We have seen a typical spinal nerve by grey and white rami communicants. So they emerge from the lateral horn cells of the spinal cord. They traverse through the spinal nerve and relay into the ganglion. They are called as preganglionic fibers. Post-ganglionic fibers, they start, and what happens is they go and relay to the effector organs. And what happens is their nerve endings, they release noradrenaline. So the fibers are adrenergic fibers. They are adrenergic fibers. Except the nerves which innervate the sweat glands and to the erector pylorem muscle, they are called as cholinergic. Only exemption. Apart from that, all other are adrenergic fibers. So they are vasomotor, which means they bring about vasoconstriction. Pseudomotor to the sweat glands, and pilomotor, so that is to the erector pylorem muscle of the skin you have. So that is called as goosebumps when you get it, is because of the contraction of this erector pylorem muscle. Okay. So that is about the sympathetic nervous system. Parasympathetic nervous activity is actually isolated and discrete, and it is actually called as the craniosacral outflow. only from the cranial nerves you can see they have certain ganglia to which it relays, and then from there what happens, from the sacral segments of the spinal cord they arise, and the ganglia are actually present on top of the viscera itself. Okay. So nerve endings are cholinergic, so they release acetylcholine only, and mainly they are responsible for conservation and energy during rest. Parasympathetic, so like it decreases the heart rate and it induces digestion. So it is actually discrete and isolated, that is the parasympathetic nervous system. So this table gives a comparison of the sympathetic and parasympathetic. So if sympathetic system is activated, your pupils dilate, your mouth gets dry, you sweat a lot, your respiration rate increases, your heart rate accelerates, inhibition of digestion, and it secretes hormones. Your adrenal gland comes into action. Here, contraction of pupils, salivation increase which is necessary for digestion, skin is dry, you don't sweat a lot, your respiration is normal, your heart rate slows down, parasympathetic slows down your heart rate, and digestion acts so all the sphincters are closed. So that is about the sympathetic and parasympathetic nervous system. Finally, we will see little about the degeneration and regeneration of neurons. Now, I am discussing very briefly about this anterograde degeneration. When the nerve degeneration occurs from top to bottom in a downward direction, if the degeneration is in a reverse upward direction towards the nerve cell body, it is actually called as the retrograde degeneration. Chromatolysis, already I told you, the Nissen substance dissolves in an injury to the neurons, then the neuron swells and then dies. Transneuronal degeneration is when the degeneration effect spreads to the other neurons. So I have restricted in this class only with respect to the nerve degeneration. I am not speaking about the diseases of the nervous system, which is very vast, that we will see during the appropriate topics like Parkinsonism, we have dementia, Alzheimer's disease, so many neurons, and also by infectious agents like poliomyelitis, rabies dorsalis, syphilis. Then you also have loss of memory like dementia and all those things. So that will become very elaborate. So here only just I am describing about the nerve degeneration. So when an axon is cut, and it is also called as axonomatosis. Okay. So with this we have completed the general aspects of the nervous system, and thank you very much for your patient listening.